In this video I will show you what impact oxygen on the coral health has. Coral reefs are important resources and provide essential marine habitats for many invertebrates, fish and algae species. They provide food for marine life, suitable habitats for growth, and provide protection from predators. Due to direct and indirect human-induced environmental changes and changing environmental factors, numerous coral reefs have been destroyed and coral mortality has massively increased. In addition, corals are severely limited in their mobility, making them susceptible to hypoxia. Hypoxia is a term for reduced oxygen levels. Under such low oxygen conditions, the corals switch from aerobic to anaerobic metabolism of glucose and thus produce the enzyme lactate dehydrogenase. This allows continuation under hypoxic conditions, but is accompanied by a significant decrease in net energy production. The enzymes allanopine dehydrogenase, octopine dehydrogenase, and strombine dehydrogenase have been discovered in numerous organisms and found to be preferred to LDH in most cases during anaerobic respiration. To supervise changes in health status, enzyme assays were developed to determine hypoxic metabolic stress in corals. For this experiment, coral stumps were grown in open flow through seawater tanks. They were then assigned to a control or treatment group. To remove oxygen from the seawater, nitrogen gas was flowed through the treatment beakers, whereas only air was flowed through the control beakers. This process of bubble formation was carried out for 3 days. Subsequently, the coral stumps were crushed and ground into a powder to preserve the integrity of the enzymes. The control cups, which were exposed only to air, did not show any physical changes. However, in the corals treated by hypoxia, physical changes were observed, such as almost complete bleaching, which worsened as the experiment progressed. Furthermore, all the tissue of the corals was lost, with the polyps remaining contracted throughout the whole period. In addition, very low LDH and ODH activity was detected in the control and treated corals, but the hypoxically treated corals showed significantly higher ADH and SDH activity. This is illustrated by the following diagrams, which show LDH and ODH activity over a 3-day period. It can be clearly seen on the graphs that their activity, neither in the control sample nor in the treatment sample, increased during the period. In contrast, ADH and SDH activity increased remarkably with time in the treatment samples. The results indicate that during prolonged oxygen deprivation intervals, coral anaerobic metabolism relies more heavily on ADH and SDH activity. However, the low occurrence of ODH and LDH in anaerobic conditions do not mean that ODH and LDH are not present in corals. The hypoxic treatment was only given for 3 days in this study. However, it has been found out that when hypoxia is given for several weeks, another pathway of anaerobic respiration can be activated. This is because ADH and SDH have been found to act as primary and anaerobic metabolic pathways during hypoxia, with ODH acting only in chronic hypoxia. Furthermore, this study shows that the health of corals is strongly dependent on the concentration of oxygen. The expansion of available biomarkers allows to better define the stress level in corals and thus prevent coral bleaching and tissue loss as well as coral mortality. In this way, it is possible to sample corals from environments considered critical and analyze their activity of enzymes and consequently identify low oxygen environments. In conclusion, ADH and SDH activity assays are suitable biomarkers for rapid analysis of hypoxia-induced stress in corals and can be used in the future for analysis of oxygen deficiency and environmental effects on corals and coral reefs. Thank you for watching.